Hello, Antonio.
Hi.
How are you doing?
I am looking for transposons.
And what are transposons?
The transposons are a part of the genetic material DNA which moves through it, producing a mutation or variation.
And what are these variations for?
This variation helps the species of animal or plants to fit their environment.
And how are you going to do that?
The first thing we are going to do is to extract the DNA.
And how are you going to extract the DNA?
The first thing we do is to add the flies into the Eppendorf with a little bit of buffer.
What is the buffer?
The buffer is a substance which maintains the pH level neutral.
And then?
We smash the sample and we add more buffer.
And what do you do after that?
Then we have to take the DNA and pass from the Eppendorf to a PCR tube.
And what is after?
We have to take the sample and introduce it into the centrifuge.
That's all?
No, we have to take the PCR tube and leave it in the mini PCR to extract the DNA.
And have you found the transposons yet?
Little by little. Now we have to duplicate the DNA. I think Lydia is doing it.
Hi Lydia, how's it going?
Hi Alonso, I'm introducing this mixture with the DNA.
What is that mixture made of?
This is a mixture made of DNA polymerase, magnesium, buffer and nucleotides.
Is it all?
No, we also have to add the primers.
And what is the function of the primers?
The primers need to begin the replication.
And the DNA polymerase?
It's a protein that moves the nucleotides in order to form a copy of the DNA chain.
Why is the magnesium needed?
Because the polymerase can't work without it.
Just that? Only add the mixture and the primers to the DNA?
No. Now we use this machine to heat and cool down the samples. Every time we do it, we get more and more copies of the genetic material.
I think that I heard Gonzalo talking about something like electrophoresis. What is it about?
I think it's the last part of finding the transposons.
What are you waiting for? Let's go and ask him.
Hi Isabella, are you doing the electrophoresis?
Right now I'm not doing it, but I can show you some pictures to explain it.
Okay, what is this picture about?
This picture is the electrophoresis.
Now, how do you know where the transposons are?
You have to see if the DNA chain is big or small.
So, the one who moves more is the biggest, right?
That is wrong. The one who moves more is the smaller because can pass through this liquid.
So the one who moves less have a transposon because it's bigger.
You're right, David, with deduction. When you see that the DNA is bigger, it can be a transposon, but not always, because the sample can be contaminated.